voltage-gated ligands and voltage-gated channels versus ligand-gated channels. Sorry about that. And what happens is when you have a voltage-gated site, you've got a slight depolarization of the membrane. This channel allows ions to go either out or into the membrane. And that continues, this depolarization travels a distance along. Eventually, it will go down into the T tubules. The depolarization, once it leaves its immediate site, which is the synapse, the site where the nerve terminal button meets with the sarcolemma. Once it goes beyond that site, it has to basically ignite an action potential in the sarcolemma. The action potential is a particular stimulus of depolarization, and that travels down the length of the sarcolemma, and wherever it comes in contact to a T tubule, travels right down the T tubule, acts on the sarcoplasmic reticulum to initiate the release of calcium ions. So as you can see here, phase 1 muscle fiber is stimulated by the motor neuron. The action potential that was traveling down the entire length of the axon of the neuron comes to the end point, which is also called the neuromuscular junction. Particular neurotransmitter, acetylcholine, was released. That's what ACH stands for. ACH will bind to receptors on the sarcolemma. These are ligand-gated channels. As a result of the neurotransmitter binding on these little channels, they will open up and allow ions, and this is where we get the term ion permeability of the sarcolemma changes. Basically, ions will pass either inward or outward, and you will have this local change in the membrane voltage. Now, if you get a little fuzzy about polarization and membrane polarization, review pages 79 and 80. We talked about this in one of the earlier chapters. I believe that was chapter 3. Once you have local depolarizations and end plate potential, as it's referred to, this will travel outside the immediate area of the neuromuscular junction, initiate an action potential. This is where we move down into the next phase, the excitation contraction coupling. The AP, action potential, will travel across the entire sarcolemma, travels along the T tubules, acts on the sarcoplasmic reticulum on each side, initiates the release of calcium. The calcium binds to troponin. The troponin moves the tropomyosin away so that the myosin binding sites on the actin are now available. The myosin head begins to bind to the actin and contraction begins. Now, this is what you see here on the diagram. You have the action potential travel down the length of this axon to the axon terminal, and you have these vesicles filled with the neurotransmitter, acetylcholine. When the action potential travels down here, it causes voltage-gated ion channels to open up. As the calcium floods in, these vesicles fuse. This is an exocytotic method. Remember we talked about that? And you're going to release these little packets of acetylcholine. This is called the synaptic cleft, the little tiny space, about 50 nanometers in spacing. And as acetylcholine travels across, it's going to attach to these particular ion channels. They will pop open. Sodium, which is in greater concentration outside the cell, will go in. Potassium will go out. This creates a depolarization here, which travels out to the rest of the to the rest of the sarcolemma. Here, we start changing the membrane voltage potential. And what you see is changes. You have sodium ion channels. Basically, this is where you have the voltage-gated ion channels. They will pop open, allow sodium to rush in, and then eventually potassium will rush out, subsequently later to restabilize the charge, but this is where you get the generation of the action potential. It travels down the length of the sarcoplasm. As it travels down the length of the sarcoplasm, what happens is it comes into T tubules. The depolarization will continue along the sarcolemma, but it will also go down into the T tubules, and the T tubules will act on the neighboring sarcoplasmic reticulum on each side. What happens then is calcium is released. This, remember, is called the triad, consisting of a T tubule, and on the adjacent sides of it are terminal cisternae. Think of it as terminal dump sites or reservoirs that will hold calcium ions until stimulated. Then they will release it onto the sarcomeres. And now we get into the sliding filament process. It's also referred to as the sliding filament theory of contraction. This was developed in 1954 by Hugh Huxley. During the contraction, thin filaments will slide past the thick filaments, so the actin and myosin filaments overlap to a greater degree. Now, we've talked about this to a degree, but let's talk about it from this point here. I'll start right here, cross bridge formation. Here is the myosin head. Now remember, the myosin, the entire length of myosin is consisting of long, thinner filaments that come to the end with two myosin heads. The myosin heads have an ATP binding site that they will undergo hydrolysis. It also has on each of the heads a site that will bind to the binding site on actin. Once tropomyosin has been pulled away, and that's done by troponin in the presence of calcium, it pulls it away. The sites are now active. You have the myosin. It will basically attach. As it attaches, it converts, or has already converted, the ATP to ADP, remember, and we have that inorganic phosphate, that's the PI. What happens, though, is the proteins are flexible, remember that. And so the flexing head will flip, and as it flips configuration, ratchets forward, it will pop out ADP, adenosine diphosphate, and the PI. You will have, the only way to attach this head, or excuse me, detach the head from the actin filament is the placement of another ATP molecule. And so that's where you have the cross-bridge detachment. Now, to get this clicked back into position to attach to the next actin, you have to have the ATP be hydrolyzed, meaning that it will be broken from adenosine triphosphate to adenosine diphosphate with a small phosphate, inorganic phosphate available. It will now be ready to basically attach again to the actin, the next nearby actin subunit with its exposed actin binding site. Okay? Now, the binding and contraction, there is a nice focus referring to the cross-bridge cycle. I encourage you to take the time to review reading it. Here you see up close, just a quick review, you have calcium binding to troponin. Troponin will pull away tropomyosin so that each and every one of the actin binding sites are freely available for the binding of the myosin head to that actin subunit. Remember, actin makes up the thin filament, myosin makes up the thick filament. At this point, the binding, you have ADP and PI still in that secondary binding site. For it to ratchet forward, when it ratchets forward, out pops the adenosine diphosphate and the inorganic phosphate, the phosphate that was broken away. So you have basically the myosin moving in one direction and the actin moving in the opposite direction. Now, that's the working or power stroke. How do we detach it? ATP will now attach to the myosin head. It will pop off. Notice it's still in a semi-cocked position. It has to be ready to do this again. How do we get it to reset itself? By hydrolysis of the ATP. So now it's ADP with inorganic phosphate ready to bind again to now another actin subunit. This brings us up to a couple of points that I want to go into in a little bit about resting potential and such. But before I do that, let me just go back here for a second, because what I wanted to do is make sure that everybody is clear on several things. One, ATP is absolutely essential for this process. And the only way that you're going to get lots and lots of ATP is to have lots and lots of mitochondria present, which will help take glucose and convert it into the maximum number of ATPs possible, but that's only done in the presence of oxygen. At any one time, some of the myosin heads are attached to the actin filaments. Sorry, there's a typo there in the notes, but don't worry about it. There's a term called rigor mortis. Maybe some of you have heard. It's called death rigor. This is where muscles begin to stiffen 3 to 4 hours after death. The peak rigidity occurs at 12 hours. It eventually begins to dissipate over the next 48 to 60 hours. This is because the dying cells cannot stop the influx of calcium from the extracellular fluid. The problem is there's no oxygen, which means ATP synthesis stops. So what happens is, remember, ATP is necessary for the cross-bridge detachment. So when rigor mortis sets in, that's where you have the muscles that are rigid. They're not going to relax. They're not going to shift in any position, because basically you've run out of oxygen. You've stopped providing oxygen. You've run out of ATP. And so a certain number of the myosin heads will be fixed and attached to the actin filaments. Why does rigor mortis decline? That's because as the muscle proteins are beginning to be broken down so many hours after death. And this is something to help you understand that, you know, you've heard of these places, or if you're aficionado of these crime programs, and they have the medical examiner and they say, well, how long has he been dead? They're actually training facilities that teach medical pathologists and criminal investigators. People donate the body, and then the body is basically provided in these cages where animals can't take away the remains. I know it sounds kind of gross, but it's basically, how do you make an estimation of someone's death? There are a variety of means. First one is, has rigor mortis set in? Then you know that it's been so many hours. If rigor mortis has been lost, then you know it's been so many hours later. Another means to tell if it's relatively recent is the core temperature of the body. And doctors will usually, or the ME, the medical examiner, will take a thermometer and they will put it into, to get the core temperature, they put it right into the liver, okay? And then they have a mathematical formula based on what's the outside temperature and what was the body temperature and what is the recorded deceased temperature, and they can tell how many hours it's taken as the body will release heat and eventually assume room temperature. Now, by the way, what I meant by those cages, etc., what if you have somebody who's dead well beyond so many weeks and their bodies have begun to putrefy? Well, they actually teach these individuals the certain signs, even in some cases, if fly larvae have begun to be present or flies have put the eggs into the nasal canals or mouths or something, that's how they make an estimate. Okay, it's been 2 weeks since the person died. This is how they do these things, and this is all based on biology and the biochemistry. Okay, now from here we're going to move into the action potential. I would encourage you to take a review of the focus part, events at the neuromuscular junction, because this will help you. Also, again, review chapter 3, pages 79 to 80 on resting membrane potential. That'll help you keep in mind. Keep in mind that on average, outside of